In the ongoing debate surrounding DNA contamination in medical products, new findings have emerged that could have significant implications for public health. The data, presented by a Canadian virologist, raises questions about the presence of specific DNA sequences in widely distributed medical vials and their potential impact on human health. While the scientific community remains divided on the implications, the claims made in this analysis have sparked discussions about safety regulations, potential long-term risks, and the broader implications of genetic interference in medical treatments. At the center of the controversy is the detection of SV40 sequences within certain medical vials. SV40, or simian virus 40, has a controversial history in medical research, dating back to concerns over its potential link to cancer. Although its presence in the current vials does not indicate the virus itself, the specific sequences identified could interact with human genetic material in ways that are not yet fully understood. The findings suggest that these sequences may inhibit the function of a crucial gene known as p53, which plays a fundamental role in suppressing tumor formation. The p53 gene is one of the most extensively studied tumor suppressor genes in human biology. Often referred to as the guardian of the genome, it acts as a cellular defense mechanism by detecting DNA damage and either repairing it or triggering cell death if the damage is too severe. When this gene is compromised, the body loses a critical safeguard against cancer development. If the presence of SV40 sequences can indeed interfere with p53 function, as some researchers suggest, the implications could be profound. Compounding concerns over genetic interference is the method of delivery for this DNA contamination. In earlier studies of DNA contamination, scientists based their safety thresholds on the assumption that the DNA in question was free-floating and exposed, meaning it would have a relatively low probability of integrating into human cells. However, the latest findings indicate that the contaminated DNA is encased within lipid nanoparticles. These nanoparticles act as a delivery mechanism that facilitates easier entry into human cells, making it significantly more likely that the foreign genetic material will be absorbed. Experts argue that if DNA wrapped in lipid nanoparticles enters human cells at a rate up to 100 times higher than previously estimated, the established safety thresholds may be vastly inadequate. This has led to renewed scrutiny over regulatory guidelines and the standards used to evaluate genetic contamination in medical products. The red lines marking the allowable DNA contamination levels in official documentation were established under the assumption of naked DNA, not the enhanced delivery provided by lipid nanoparticles. Beyond inhibiting p53 activity, SV40 sequences have also been implicated in the process of DNA integration into the human genome. The possibility of foreign DNA incorporating itself into human genetic material has been a long-standing concern in gene therapy and genetic medicine. The scientific process known as reverse transcription, wherein genetic information is copied into human DNA, raises the question of whether long-term genetic alterations could be taking place as a result of these exposures. If integration occurs, the presence of SV40 sequences within the genome could lead to continuous production of SV40-related proteins in human cells, potentially exacerbating the suppression of tumor-fighting genes over time. This could create a scenario in which the body gradually loses its natural ability to prevent the development of abnormal cell growth, leading to an increased risk of cancer years or even decades after exposure. One of the most striking elements of the recent findings is the significant variation in DNA contamination levels between different vials sourced from various countries. Samples obtained from Ireland, Canada, and Australia showed differing amounts of genetic material, with certain batches demonstrating particularly high concentrations of SV40 sequences. The question remains as to why such discrepancies exist and whether manufacturing inconsistencies are contributing to the contamination levels. The presence of spike protein encoding DNA in these vials also raises additional concerns. While the primary function of these DNA fragments is to instruct human cells to produce a specific protein, their high concentration in medical products raises questions about long-term effects and regulatory oversight. If these sequences are entering human cells at an enhanced rate, the possibility of prolonged expression of spike proteins or unintended genetic effects must be carefully evaluated. Despite the significance of these findings, the response from the scientific publishing community has been notably restrained. Several leading journals have reportedly rejected the study without sending it for peer review, raising concerns over potential censorship or hesitancy to engage with controversial topics. The reluctance to engage with such data, despite the potential public health implications, has only fueled speculation and skepticism. The history of medical research is filled with instances in which early concerns were dismissed, only to be validated years later through more rigorous studies. The rejection of data without full scientific evaluation risks delaying important discussions that could shape future safety regulations. While the data presented may still require further verification, the potential implications warrant serious investigation rather than outright dismissal. Critics of the findings argue that the presence of SV40 sequences does not necessarily equate to a direct cancer risk. They emphasize that while SV40 was historically linked to tumors in certain experimental settings, its role in human cancer development remains uncertain. Furthermore, they point out that the immune system has mechanisms for dealing with foreign DNA, making integration into the genome a less frequent occurrence than some fear. Others argue that even if the contamination levels exceed established thresholds, the absolute risk remains low when considering broader epidemiological data. Given that medical treatments undergo extensive clinical trials, proponents of existing safety standards suggest that if these DNA fragments were having a widespread negative impact, significant increases in adverse events would have been observed by now. Yet the core of the argument presented by those raising the alarm is not that a catastrophic health crisis is imminent, but rather that safety standards must be reassessed in light of new data. The interaction between SV40 sequences and human DNA, particularly when delivered via lipid nanoparticles, represents an area of study that has not been fully explored. Until rigorous independent research is conducted, it remains unclear whether current assumptions about genetic safety hold true in this new context. A major factor in this debate is the broader issue of trust in regulatory agencies and scientific institutions. Past controversies over medical safety have led to heightened skepticism among the public, and any perception of withholding information or dismissing concerns outright only serves to deepen this mistrust. Transparency, open dialogue, and independent verification of data are essential in maintaining public confidence in scientific and medical advancements. As the discussion continues, several key questions remain unanswered. What is the long-term impact of DNA contamination in medical products, particularly when delivered through lipid nanoparticles? Are current regulatory thresholds sufficient, or do they need revision to reflect modern delivery mechanisms? Should independent research institutions be tasked with replicating these findings to provide further clarity on the risks? The issue at hand is not simply a debate over laboratory data but a question of how scientific institutions respond to emerging concerns. If the risks associated with genetic contamination are confirmed, addressing them early could prevent long-term health consequences. Conversely, If further research disproves these concerns, openly engaging with the data and providing clear explanations will help reinforce trust in medical science. Regardless of the outcome, one thing remains clear. The conversation surrounding DNA contamination, genetic interaction, and long-term cancer risk is far from over. Scientific inquiry must continue, free from bias or suppression, to ensure that public health decisions are based on the best available evidence. Only through open and rigorous investigation can the true impact of these findings be understood, and appropriate measures taken to safeguard future generations. If you found this information insightful, don't forget to like and share this video to help spread awareness. Let me know your thoughts in the comments. Do you think these concerns deserve more attention? Stay informed, stay curious, and subscribe for more updates on the latest in health and science. Thanks for watching.